Hello and welcome to today's webinar at Microscopy Focus, which is a Leica Microsystems production powered by Bite Size Bio. Today's presentation is titled Live Cell Fluorescence Lifetime Multiplexing Using Organic Fluorophores, and it's being presented by Dr. Michelle Fry. Dr. Fry completed her PhD studies in the lab of Professor Kai Johnson at EPFL in Lausanne and MPI for Medical Research in Heidelberg. Her PhD involved synthesizing and characterizing photoactivatable probes for super resolution microscopy and engineering the self-labeling protein tag HALO tag 7 for fluorescence lifetime multiplexing. She continued her work in fluorescence lifetime multiplexing using synthetic organic probes. Michelle is currently a postdoctoral scholar at UCSD in the lab of Professor Jin Zhang, where she works on fluorescent biosensors for application in super resolution microscopy. After the session, I'll be putting your questions to Dr. Fry, so please type your questions as they come to you into the questions box, which appears on the left of your screen. So now, over to you, Dr. Fry, for the presentation.
Thank you very much for this kind introduction. It's a pleasure to be here and talk to all of you. As already has been said in the introduction, I did my PhD in the lab of Kai Johnson, and now I'm a postdoc in the lab of Jin Zhang. And today I will talk to you about my work that I did in the lab of Kai during my PhD, which focused on the use of organic fluorophores for fluorescence lifetime multiplexing in living cells. To start off this presentation, I would like to give a brief introduction about multiplexing in fluorescence microscopy in general. So whenever you're about to multiplex several targets in your sample, you will label them with multiple different fluorophores. And most importantly, they are different. So they have to be distinguishable by one of their photophysical properties. I brought you here a list of the most important photophysical properties of fluorophores. And once you go through that list, you might figure out that probably the first properties, so the spectral properties of a fluorophore, are the ones that are most commonly used to multiplex fluorophores. We commonly use the excitation and emission spectra of fluorophores to separate them from each other. How is this commonly done? So we use a concept called spectrally resolved detection, in which we choose fluorophores that have excitation and emission spectra that are well separable from each other. And so we can collect the emission of the different fluorophores in different spectral windows. This works really, really well up to 4 fluorophores. And probably a lot of you have actually done something like that before. However, this process is also associated with several challenges. If we want to multiplex more than the 4 fluorophores, we start to run into problems because the fluorophores have broad spectra, both excitation and emission spectra. This causes crosstalk between the different fluorophores and necessitates a lot of control samples and unmixing of the different fluorophores. Ultimately, the spectral space is also limited, so we cannot multiplex an indefinite number of fluorophores with each other, making the system limited. So if we want to go beyond that and multiplex more fluorophores, we have to turn our attention to another property up in that list. And many different people have worked on different strategies. And the one property that I turned my attention to was fluorescence lifetime. So how can we use fluorescence lifetime to multiplex multiple targets with each other? In fluorescence lifetime multiplexing, it is our goal to label our sample with fluorophores that are spectrally rather similar or even identical so that they cannot be distinguished by their spectral properties. When we then measure their fluorescence lifetime information by FLIM microscopy, so fluorescence lifetime imaging microscopy, the 2 fluorophores that we're using should be distinguishable by their decay patterns. We can then use fitting or pattern matching to assign to each and every pixel in our sample what the contribution is between the 2 fluorophores that we have to stain this hypothetical sample here. And we can now distinguish the 2 components by their lifetime. An even easier approach to do the separation is by using the so-called phasor plot. The phasor plot is nothing else than the Fourier transform of the decay curves you have seen in the previous slide. In the phasor plot, pixels that have similar fluorescence lifetime show up as these clusters. And in phasor space, we can make use of very simple math based on vector addition rules to separate which pixels contain which contributions if we have 2 or maybe 3 components present. Having learned about this technique, I asked myself which fluorophores and which labeling strategies would be ideal to be used with this microscopy method. And that's when I turned my attention to so-called organic or synthetic fluorophores. These fluorophores are extremely bright and also very photostable, which makes them very useful tools for live cell applications. They're also available in many different colors, especially in the far red region, where fluorescent proteins are not abundantly available. And we can have cell permeable versions available so that we can really perform live cell experiments. I then asked myself, which labeling system should I use? How should I target my fluorophores to the places that I want to look at? And the 2 things that I focused myself on was, on one hand, the self-labeling protein tags, and on the other, synthetic probes, which are fluorophores that are attached to a so-called targeting ligand that will bring the fluorophore to the places of interest. In today's talk, I will tell you 2 stories. First, I will focus on how I used self-labeling protein tags to create fluorescence lifetime contrast. And then I will tell you how we can use fluorescent synthetic probes for fluorescence lifetime multiplexing. Ultimately, you will see how the 2 of them come together to take advantage of the benefits of both of them. So let's get started with the self-labeling protein tag approach. And to start this section off, I would like to bring everybody onto the same page and briefly introduce to you what the self-labeling protein tag is. So self-labeling protein tag is a protein that can covalently and very efficiently react with a specific ligand. In the case of this example here, where we have the self-labeling protein tag halo tag 7, this ligand is the chloroalkane that you can see here. The protein is going to react at this position here at the chlorine, and on the other side of the chloroalkane, we can attach a fluorophore, which is some sort of a cargo that will then, after the reaction has proceeded, be attached to our self-labeling protein tag. This then renders the halo tag fluorescent, and we can use it analogously to a fluorescent protein. So we can express it as a fusion protein to our target of interest. You might ask yourself now, but how do we generate lifetime contrast with this kind of system? Isn't that just a labeling system? And the devil here lies really in the detail. So fluorescence lifetime of all kinds of fluorophores is influenced by the local environment of the fluorophore. It's really the specifics of the local environment that determines what the precise fluorescence lifetime is going to be of the fluorophore. And in our case, the fluorophore is located on the surface of the self-labeling protein tag, meaning that the local environment is the protein surface, and the protein surface is now really key to determine what fluorescence lifetime we're having. I made use of this concept and actually engineered the halo tag in a way that I would gain new halo tag variants that have different fluorescence lifetimes. So I first had a look at the crystal structure and identified residues that would be close to our fluorophore. I then mutated them, and over several rounds of screening, found 3 new halo tag variants that by now are named halo tag 9, 10, and 11. And all of them have different fluorescence lifetimes when compared to halo tag 7. So halo tag 9 has a longer lifetime, and halo tag 10 and 11 have shorter lifetimes. To illustrate this a little bit better, I brought you the following plot. In this experiment, I tested 6 different fluorophores in different spectral regions on all of our 4 halo tags. And you can see from this plot, which is really nicely colorful, that we can really cover a broad range of different fluorescence lifetimes, ranging from something like 1 nanosecond up to 4 nanoseconds. Here, I would like to highlight 2 fluorophores that have also been developed in the lab of Kai Johnson, MUP555 and MUP618. For both of the fluorophores, we actually generate an evenly spaced gradual distribution. So for MUP555, we're going from around 1 nanosecond up to 3 nanoseconds. And for MUP618, we're covering a range from 1.5 up to 4 nanoseconds. We can then use these values and actually calculate the differences between the different halo tags for the two fluorophores. And as you can see from these plots, for both of them, the combination of halo tag 9 and halo tag 10 creates the largest lifetime difference of up to 2 nanoseconds for MUP618 and around 1.5 nanoseconds for MUP555. This makes these combinations extremely promising for fluorescence lifetime multiplexing. We can also look at the phasor plot and see how the different clusters of the different protein tags behave in the phasor space relative to each other. Then, before actually proceeding to fluorescence lifetime multiplexing, we wanted to answer one last question. And that question was whether the fluorescence lifetime could be influenced by the target where we're going to bring it to in the cell. So we generated a lot of different fusion proteins of all of our 4 halo tags, targeting our halo tags to different subcellular locations and different proteins. We used things such as the nucleus via histone H2B, or lysosomes, LAMP1, the mitochondria, SEP41, which is a microtubule binding protein, and we had a look at the fluorescence lifetime. As you can see from this plot is that throughout one halo tag, the fluorescence lifetime seems to be rather stable, and the target itself, or the protein that we're going to fuse it to, does not have too big of an influence. Obviously, there's always some outliers. So it's still a good idea if you were to use this technique to create your fusion and to double check what the fluorescence lifetime is. But overall, it is pretty predictable what the fluorescence lifetime is going to be. Knowing this, we set out to perform our first fluorescence lifetime multiplexing experiments. In this case, I labeled the mitochondria via a halo tag 9 fusion and the nucleus via a halo tag 7 fusion. I then labeled the cells with MUP618 and proceeded to flame microscopy. If we just measure the intensity of the fluorophores, we cannot distinguish them. But as soon as we start to acquire the lifetime information and use the phasor plot to separate the two components, we can nicely see where the mitochondria and the nucleus is located, and even see those mitochondria that are going above and below the nucleus. Having done this with MUP618, I also wanted to see if it also works with MUP555, because that fluorophore also had nice differences in fluorescent lifetime. And indeed, this also works with MUP555. Here I'm showing you a combination where I used halo tag 10 and halo tag 9, staining the mitochondria and the microtubule network in U2S cells. The system doesn't only work with combinations of 2 halo tags, we can also combine 3 of them together. So we tried doing that with different combinations. Here I'm showing you an example where we combined halo tag 7, staining the nucleus, halo tag 9 attached to TOM20, which is localized in the mitochondria, and halo tag 11 highlighting the Golgi apparatus. We can then separate the 3 of them using the phasor approach, and we can nicely see the 3 components in relation to each other. At this point, I would like to give a brief summary. So this system has many advantages. The halo tag is very versatile, and we can fuse it to many different proteins, so we can target many different targets. The lifetime differences we are creating with the series halo tag 7, 9, 10, and 11 is pretty big. So the biggest difference we can see is almost 2 nanoseconds. And as I've shown you previously, the lifetime does not depend too much on the fusion protein, so the system is very predictable, which is good for us if we're trying to switch around the targets. However, this system has one main disadvantage, and that disadvantage is that it relies on the expression of 3 targets. this can either create the situation that we need to work with extremely big constructs, because we would just concatenate all of them using, for instance, self-cleaving peptides, so-called T2A and P2As in between. And then that gives us the trouble with working with these rather big constructs. If we want to circumvent this, we could potentially also generate stable cell lines and then transiently transfect a second component on top of that. However, that is rather time consuming, and maybe you don't want to generate a stable cell line for all the targets that you want to look at. Also, it is rather difficult to tune the expression level of the individual components. And when you look at your sample, you might find cells that are expressing one of the components very highly and the other ones rather low. And this then generates some differences in the relative brightness of the two components, which can make it tricky for FLIM multiplexing. And last but not least, certain cell types might just not be very easy to transfect, and that could prevent you from doing such an experiment. So that's when I asked myself, is there any trick? Can I circumvent these challenges somehow? And that's when I turned my attention back to what I mentioned to you in the beginning. I wanted to use synthetic organic probes instead of the self-labeling protein tags in order to generate lifetime contrasts. So what are these probes? So organic probes are molecules which consist of a fluorophore part, this one here, or the silicon rhodamine here, and they are attached to a so-called targeting ligand. Targeting ligands fall into two classes. You can either have so-called accumulating target ligands, as for example, this group here. This group here consists of a tertiary amine that can be protonated in case it is present in very low pH. And this probe is staining the lysosome. So as soon as this probe enters the lysosome, this tertiary amine will get protonated. And once it is protonated, the probe cannot leave the lysosomes anymore because it becomes cell impermeable. That means that the probe will accumulate over time and renders this probe a very good lysosomal stain. On the other hand, we can have binders as targeting ligands, which really bind to specific sites on proteins or on biopolymers. I brought you here an example of a binder that is going to bind to DNA and is an effective DNA stain. Overall, with these organic synthetic probes, we're covering again the same advantages as with the fluorophores that we use for halo tag labeling. They are bright and photostable, available in many colors, and are available in cell-permeable versions. But in addition to those benefits, we now do not need any transient transfection or any stable cell lines, because these targeting ligands are actually going to make our probes go to the places that we want them to go. Also, those probes can be very easily combined. I can just choose before I do the labeling which of the probes I want to combine with each other, making diverse experiments very, very simple to carry out. However, there's one last question, which is actually quite crucial for what I want to do. So I didn't know before I started, do those fluorophores actually show any lifetime differences? And unfortunately, only very few of those probes have been characterized with respect to their fluorescence lifetime. And that's why at the beginning of my work on these probes, I just simply characterized a whole bunch of them because we didn't know how they would behave. So how did I characterize those probes? I used U2S cells and stained the U2S cells with all of those probes individually. I then acquired FLIM microscopy images and used the phasor plot to first assess the homogeneity of the lifetimes of these probes. If I would see multiple clusters for one probe, that would not be a good indication because this probe would not be suitable for fluorescence lifetime multiplexing. And I sorted the few probes that showed this unfavorable behavior out of my list of fluorophores. The ones that showed homogeneous behavior were then fitted to a mono B or 3 exponential decay, allowing me to obtain an average fluorescence lifetime for all of them. I've done that for 18 probes, which belong to 5 spectral regions, and I've also done this for 3 different cell lines to check whether there would be a huge variability between different cell lines. Next, I grouped the fluorophores into these 5 groups by measuring their excitation and emission spectra, grouping fluorophores together that have very, very similar spectra. Once I had set up those groups, I overlaid all the phasor plots of the probes that belong to the same group. Here I'm showing you 2 examples. On the top, you can see 3 probes, SIR-DNA, SIR-actin, and SIR-tubulin. However, very unfortunately, all of those probes have an overlaying cluster, meaning that the fluorescence lifetime of all 3 probes is extremely similar and therefore not suitable for fluorescence lifetime multiplexing because separation would just be too tricky or almost impossible. On the bottom, I brought you a different example of 2 probes that actually show quite a big difference in fluorescence lifetime. Rhodamine B, which is a mitochondrial stain, and SPY55 actin, SPY55 DNA. Once I identified the probes that actually do show a difference in fluorescence lifetime, I also calculated the difference in fluorescence lifetime and reported them in these kind of plots here. From these plots, you can very nicely and easily find out which combinations are promising to perform fluorescence lifetime multiplexing. The combination we saw here on the left, for instance, rhodamine B and SPY55 DNA has a lifetime difference of 0.75. And the combination that showed the highest lifetime difference in this orange spectral region is the combination of SPY55 DNA and SPY55 actin, here with 1.2 nanoseconds. Having identified these very promising candidates or pairs of probes that are promising for lifetime multiplexing, I then set out to perform this kind of fluorescence lifetime multiplexing in each of the different spectral channels. So here I'm showing you an example in the green spectral region where I used mitotracker green and lysotracker green to stain the mitochondria and the lysosome. Upon measuring of the intensity, again, we cannot differentiate which probe is where, but as soon as we measure the lifetime information and use the phasor plot again to perform the separation of the 2 components, we can now nicely observe the relative position of the lysosomes and the mitochondria. The Leica SP8 microscope that I used to perform these measurements also has a very cool feature called the fast film image, which is given to you on the fly when acquiring the data. The fast film image reports on the average photon arrival time, and it doesn't require any fitting or phasor analysis. It just tells you by this kind of color scale where we have pure components, such as here in the mitochondria, or overlapping areas here, these yellow areas, or pure lysosomes here, these red dots. This is a very convenient tool to have a fast inspection while performing the microscopy and to get a rough estimate how well the separation will work. I could not only do this kind of separation in the green spectral region, but as I've previously shown to you, for instance, in the orange spectral region, the combination of rhodamine B and SPY55 DNA was a promising combination. So we also did that for a few different combinations in the orange spectral region, as well as in the red spectral region and the near infrared. Overall, I found 8 pairs in 4 spectral channels, so the green, orange, red, and near infrared region that could be multiplexed in this fashion. Unfortunately, in contrast to the halo tags, it was not possible to find combinations of 3 probes that would allow to separate 3 of them at the same time. The lifetime differences there were always just too small, so that didn't work very well. However, in order to push the multiplexing to a higher degree, we then set out to perform a combination of spectrally resolved detection and fluorescence lifetime multiplexing. So how did we do this? We simply chose 2 of the spectral channels. In this example here, the green spectral region and the red spectral region. And in each of those channels, we chose 2 probes that could be multiplexed by lifetime multiplexing. What we did then is that we acquired 1 image in the green spectral region and 1 in the red spectral region and used the lifetime information to separate the 2 components, giving access to a 4-component image in the end. So this allows us now to look at the nucleus, the F-actin network, mitochondria, and the lysosomes at the same time. So let me ask a question here. What if I now want to visualize a target for which there is no probe? I think most of you have seen a lot of mitochondria, a lot of lysosomes by now, but what if I want to look at the protein that is a little bit more special for which there is no probe? Or what if I do have a probe, but the lifetime difference is not suitable or not big enough in comparison to my other probes? That's when I would turn my attention again back to the self-labeling protein tags, because they have very predictable fluorescence lifetimes, and they can be technically combined with these probes. So to check if that really works, I expressed the halo tag as a fusion protein with calreticulin and a cadal targeting sequence, which brings the halo tag to the ER network. And as you can see from these images here, when I stain the halo tag here with the fluorophore MUP555 and combine it with a probe SPY55 DNA, I can nicely multiplex the nucleus and the ER using this combination. This now gives me access to a target that I could previously not image. Instead of using the halo tag, I can perform the same imaging also using a snap tag. The snap tag is a related self-labeling protein tag, which is orthogonal to the halo tag. And in this example here, I used a similar fluorophore, MUP555BG, which carries the specific ligand for the snap tag, and I multiplexed it with SPY55 actin. So we can look at the actin network and the ER at the same time. Having this in hand, I could now also expand the degree of multiplexing we've done in this combination of spectrally resolved detection in combination with fluorescence lifetime multiplexing. Before, I've shown you a 4-component image, but once we wanted to go to 6-component, we were actually limited because the probes that we've been previously using were redundant with regard to their targets. Now, if we introduce a halo tag, we can actually obtain a 6-component image combining 5 probes for lysosomes, mitochondria, F-actin, DNA, and microtubules, and combine them with MUP555 chloroalkane, which is going to label a halo tag that is localized to the ER, and we can get access to this very beautiful 6-component image. You might ask yourself, did we stop there? Did we leave it at 6 components? No, we didn't, because over here you can see there is some spectral space that is still free. And using the halo tags, so 2 halo tags, indeed halo tag 7 and halo tag 11, in combination with a snap tag. So now we're having 3 self-labeling protein tags and 5 probes. We can now image a so-called 8-component image, where we're looking at the lysosome, the mitochondria, the plasma membrane, the actin network, the ER, the Golgi, the nucleus, as well as the microtubule network. This was really, really cool to see, and we could essentially double the number of accessible targets with this kind of experiment. Last but not least, all of the experiments that I have shown you today were performed in living cells, but so far I've only shown you still images. Because we measure in living cells, we can also record multiple frames after each other and technically generate a video. This is exactly what you can see here. So I brought you some still images here where you can see a lifetime multiplexed image after every 2 minutes. And we can see how the mitochondria move around, how the microtubules rearrange a little bit. Unfortunately, this technique still doesn't have the most or the best time resolution. We need to acquire the 4 or the 3 different channels, in this case, after each other. So that limits our time resolution a little bit. With this, I'm at the end of my presentation. So I hope I could convince you that these organic synthetic fluorophores are really convenient tools to perform fluorescence lifetime multiplexing. And I hope I could show you how we can use 2 labeling strategies, self-labeling protein tags on one hand, and the synthetic probes on the other hand, to perform this kind of microscopy. I would like to remind you at this point that the 2 of them have different advantages that might be complementary to each other, but can also be combined. So self-labeling protein tags can be used for many different targets. They have to be proteins. And because the lifetime differences do not depend too much on the fusion, we really know what the lifetime differences are going to be. On the other hand, if you're going to work with the probes, you will not need any transient transfections or stable cell lines, which makes your experiments a little bit more rapid and easier to perform. And the probes can be really easily combined with each other, so you can try many more combinations in a shorter amount of time. So overall, fluorescence lifetime multiplexing can expand the number of accessible targets. It can do that by a factor of 2 or even 3 if we find groups of 3 probes that can be multiplexed together. With this, I'm at the end of my presentation, and I would like to thank a few people. So I would like to thank the whole Johnson group for spending all their time with me during their PhD, and I would like to in particular thank Julien Hiblot and Birgit Koch, who really helped me throughout those 2 projects. I would obviously also like to thank Kai Johnson, who gave me the opportunity to work on these really exciting projects. I would also like to thank the collaborators, Mirek Tarnavski, Julia Roberti, and Elisa Deste, who also supported me on this journey. Last but not least, I would also like to thank the funding. So I was supported by the Max Planck Society, EPFL, and an SFB Transregio grant from the Deutsche Forschungsgemeinschaft. And I would like to thank you for your attention.
Thank you, Michelle. That was an excellent presentation on some really ingenious work, actually. So we have a few questions from the audience. If anyone has a question, and anyone else has a question in the audience, please feel free to post it in the questions box that appears on the left-hand side of your screen. So first up, Michelle, the first question is, what would be the ideal properties of 2 probes for fluorescence lifetime multiplexing?
So if we're going to use 2 probes to kind of separate 2 of them, it would be very ideal if this lifetime difference would be extremely large, because if this lifetime difference is bigger, we're basically more sensitive and can detect smaller changes. Another property that I haven't talked about too much today is that it is good if the relative brightness of these 2 probes is rather similar, because if one of them is really dim in comparison to the other, we're running into some problems after the separation. So if the brightness level can be balanced, that is also very ideal.
Okay. Okay, so next question. Are all channels acquired simultaneously during the imaging of 3 or more channels? And how fast is the frame rate of your acquisitions?
So unfortunately, at the moment, the channels are not acquired simultaneously, even though technically that would be possible. I ran into some trouble during the analysis if I acquired them simultaneously. So far I have done them individually. Obviously, if I could acquire them simultaneously, I could cut down on how fast this acquisition would work. But at the moment, that doesn't work for me.
Sure. Okay. We have a question asking you to go back and explain the mechanism behind the multiplex staining with halo tag. I don't know if you want to go back to that slide, or we could have just advised the person to go and have a look at the on demand. But would you like to recap that briefly, or?
I can briefly mention that. Let me just go to, I think I had a slide here.
Yes.
Yeah, so I can probably explain this on this slide here. So I generated these 4 halo tags, and when you label them with one and the same fluorophore, the fluorophore will be spectrally the same, but it will have different lifetimes. So if I want to multiplex these tags with each other, I'm going to choose, for instance, 2 of them. So I'm going to choose, let's say, the halo tag 7 and halo tag 11. And what I'm going to do is I'm going to express halo tag as the fusion of one of my proteins of interest. So let's say H2B, and halo tag 11 as a fusion protein of my other target. let's say, a plasma membrane bound target. So first I have to express the 2 halo tags in a cell. So I can do that either by transient transfection or generating of a stable cell line. Once I have these cells that express these 2 tags, I'm going to label them just with 1 fluorophore. So both of them will be labeled with the same fluorophore. But because the lifetime will be different, I can measure the lifetime information, which will differ from each other. And I can use the phasor plot that I think you can see graphics here. I can use the phasor plot to then separate the 2 components. I hope that became a little bit more clear now. Otherwise, I think you can probably also read up some more details how this is done in one of the 2 publications that I've listed here.
That was a nice quick recap there, actually. Okay, so does this method allow you to differentiate separate proteins like receptors on a cell surface, for example?
Yes, it could do that if you are able to express your receptors with a fusion of the 2 halo tags, or if you have probes available for your 2 cell surface proteins. I'm not exactly sure what you want to do, but it's possible that this works. Yes.
Okay. Next up we have, is the number of photons that you need to collect to measure a lifetime, is that a lifetime measure? Okay, hang on, this is a very... Is the number of photons you need to collect to measure lifetime a major limiting factor to have high temporal resolution with live imaging of 8 components?
Yes, so fluorescence lifetime imaging is a photon-intensive technique. So in order to do the separation, we do need to acquire quite some photons. However, the microscopes that we are using nowadays have become a lot better, and we can actually image quite quickly. It's still a limitation, but we're still a lot better than before, I think, yes.
So you mentioned the fluorescence lifetimes in 3 different cell lines. Did you find big differences there?
Yeah, that was something I was very curious about myself. So I compared 2 things. I compared between different cell lines. And obviously, I also did multiple replicates within 1 cell line. And what I actually found is that for most probes, these variations, both of them within 1 cell line and between different cell lines is rather small. But there is a few probes. It's mostly probes for lysosomes, which show bigger spread. And that's not just between the different cell lines, it's also among the same cell line sample. And the explanation I came up with for that was that lysosomes are not homogeneous species. The pH is not the same in all of them. And probably these small differences in pH, again, the local environment probably influences the fluorescence lifetime of these probes. Yeah. So it's not per se the difference between different cell lines, but certain probes just have a little bit less homogeneous properties, let's say.
Okay. Let's see. Would it be possible to multiplex 4 components by fluorescence lifetime multiplexing?
That's actually a good question. So I've shown you today how we can multiplex 2 and then eventually also 3. And I think technically it would also be possible to multiplex 4. However, when we use we specifically use the phasor approach, we run into some mathematical trouble, because if you imagine you have 4 components, you're going to have a rectangle. The solution is not going to be unique anymore. So if we want to multiplex 4 components, we either need to use different separation strategies or add another component. So add some spectral information so that the separation becomes easier. I think in general, it's good to say the more components you add, the trickier the separation goes. So 2 is very simple, 3 already becomes more challenging. And when you do 4, I think it's easier if you follow our approach where we combine some spectral information and some lifetime information.
Sure. Okay. So following up on that facts question, so the reworded question is it possible to see only differences in the general fluorescence with your technique, or does it allow us to count the receptors to quantify?
Sorry, can you repeat that once more?
So is it possible to see, basically, can you quantify the number of proteins that are fluorescing?
Okay, so at the moment, I've only performed confocal experiments where I think counting in that sense is not possible because we're not looking at single molecules. If you think about actually counting the receptors, I think what you would probably want to do is some single molecule technique. And there's some really cool work emerging right now where people actually combine lifetime information with single molecule approaches. So maybe you should one could have a look at that, but I haven't currently tried that. So that's something that is maybe a little bit more tricky.
Okay, let's see. Could the self-labeling proteins be interchanged with the probes? In other words, can I use probes where I once used the self-labeling proteins?
So the first question, or the first thing you have to do to answer this question, is you have to know which targets you want to look at, and then you have to go and check the literature and see if these probes are actually available. So the ones that I have tried here in this study that I've presented to you today, they're all commercially available. So I have tested probes that can label mitochondria, lysosomes, F-actin network, DNA, and microtubules, I think. I didn't test any others. There's some other probes out there that also stain other targets. So if that's the target you want to look at, then yes, you can replace the self-labeling protein tag with a probe. However, you might want to look at something for which there is no probe, and then you're better off if you use the self-labeling protein tag.
Okay. A couple more questions. Did you ever try to fix and embed the labeled cells? And if yes, how did the lifetimes change?
I have tried this for the halo tags, however, not with embedding. So I just fixed the cells. Mm-hmm. If I remember correctly, I did PFA fixation and the lifetimes didn't change too dramatically. Maybe a small change for the halo tags, but I didn't try that for the probes. I have to add here, so some of these probes actually work better in living cells than in fixed cells, because sometimes you lose their specificity once you fix them. So that could be a limitation there, that the probes themselves just don't really work very well in fixed cells.
Okay. The final question, unless any drop in at the last minute. Have you tried STEAD together with the multiplexing of 2 and 3 targets?
So yes, we tried that. Again, this we also tried with the halo tag approach. We tried that with 2 halo tags, and it did work. We could get a higher resolution and we could still separate the 2 components. I think I should say it still requires some more optimization. The pair of halo tags that we used back then was the pair of halo tag 9 and halo tag 7, the 2 brightest ones, because for STEAD you need really bright fluorophores. So we could maybe revisit that and actually take a pair that has a bigger lifetime difference to make the separation maybe a little bit easier. But yes, indeed, that can also work. But it's not an easy technique to perform, I have to say.
Okay. There's a further follow-up to the facts question, but I think I'll let you answer that offline. I think it might need a bit more discussion than we can do here.
Sure, I can do that.
Okay, perfect. Okay, so that brings us to the end of the webinar, and thank you again, Michelle, for a very interesting presentation and a great discussion. And thanks to you, the audience, for taking time to attend and listen in. If we did not get a chance to answer your question, please know that they will be addressed offline by email. So until next time, goodbye from all of us at Leica Microsystems and Bite Size Bio.